go ahead and go over lipid digestion. So this is, you eat some fat, hopefully it's delicious, and then you get to digest that fat, which hopefully works really well in your body, so you can continue enjoying, well, you know, deep fried chicken and stuff. So, first, in your mouth, you have lingual lipase. It is a lipase, and it breaks down lipids. So that happens in the mouth, pretty simple. Then you go down to the stomach, where you have chief cells. And so here's a feather for, I don't know, when I hear chief, I think like I am the Indian chief. So there's a feather, and cholecystokinin. Cholecystokinin. And I probably have multiple people saying there's a spelling error, but it's called cholecystokinin. And it's excreted via chief cell activation, and that gets you some bile salts coming in, and that causes emulsification of the fats. And so the fats previously were in their lipid structures as like full triglycerides. Now they're going to be monoacylglycerides and free-floating fatty acids, and they will be packaged up into micelles eventually along this process. So the next thing is what helps complete the micelle creation process, which is when it goes into the duodenum. So number 3 active thing, secretin. Okay? Secretin puts out a bunch of pancreatic enzymes, and we will probably learn that cascade in depth, I'm basically positive. But for now, know that we have panzymes, and those help cause the micelle, okay? And I'm going to move the micelle so that it's right here. Micelle is basically when you have a bubble with all the hydrophobic in the middle, and it doesn't really have a whole bunch of proteins, which is one of the things that makes it different than what we're going to talk about later, chylomicrons. It's also in the small intestine. The other thing that happens in the duodenum is you get secretion of hydroxyapatite to raise the pH. And so that's like HPO4, if I remember correctly. Okay? So there's something, there's something, there's something, there's something. There are 4 separate things going on here that you want to remember. And then you can go ahead and pause and review that if you want in your head. We're going to press on. Chylomicrons are what happened next. So it gets absorbed into the lymph, all these bits, the fatty acids, the monoacylglycerides, which are called mono because there's only one fatty acid, which is on the number 2 location. The 2 on the sides come off, which kind of makes sense. It seems like those would be easier to break off than the one in the middle that's got all that stuff around it. So you also have, as it's going into the lymph, how do chylomicrons get formed? Well, in some of the intestinal epithelial cells, you also, as it's being absorbed into the lymph, between the cells and the lymph, it gets cholesterol, and apoproteins. Okay? And those then form, this is what our chylomicrons will look like today, and that's how happy we are about it. And we have a chylomicron in the lymph, which then gets deposited into the blood. And in the lymph, the only apoprotein that you get is ApoB48. B48. At least, all right. And so ApoB48 comes before it gets to the blood. Once it's in the blood, it will also get, so that's, it will get ApoC2 and ApoE, okay? And HDL actually is the one that delivers them to the chylomicron because HDL is floating out here. And it shares ApoC2 and ApoE with the new chylomicrons coming in. So chylomicrons, all right, here I'll draw an intestinal cell that includes part of those words. So, chylomicrons, once they're in the blood, what are they doing? They're going to go around and they're going to deliver lipids, because they have a ton of lipid content on the inside. On the outside, they have hydrophilic heads and proteins. On the inside, they have all their tails. And the tails and the heads are connected, but the tails are facing inward. Okay? So they're all connected molecules. It's just the hydrophobic parts of those molecules are facing inward, and the hydrophobic is facing outward. Matter of fact, it looks a lot like a phospholipid membrane, except it's only one layer that's bubbled in on itself. All right, so chylomicrons go around and they deliver. After they've gotten their ApoE and ApoC2, they are able to deliver their lipids. They're able to deliver lipids to cells that need them, and the cells will bind, I believe it's with ApoE, and have lipoprotein lipases that cut off a bit of the chylomicron. And so you get a little bit of the chylomicron in here, and then the rest of it continues away. And it's a little bit smaller now because some of the triglycerides have been taken. Okay? And we had lipoprotein, which makes sense because it's both a protein and a lipid. So lipoprotein lipase. Lipase meaning it's a lipid enzyme, and so it's cutting it off. And then you have a smaller chylomicron going on. This repeats multiple times until you are left with what is called a chylomicron remnant, which doesn't have very much stuff left. Chylomicron remnant ends up going to the liver, where it binds with an LDL receptor. An LDL receptor, I believe it's LDL receptor 2. We don't need to know that though. But it binds with an LDL type receptor and goes into the liver, where it is then metabolized, and it goes through so parts of it, the glycerol will go through going into DAP, dihydroxyacetone phosphate, into the the glycolysis pathway. That way the fatty acids will go down to acetyl-CoA, and they will be used for things like cholesterol formation. There's actually some cool stuff in the liver that happens that I didn't notice her wanting us to know. So I'm going to press onward, I think. What do you think? Would you like to know all of it? Yeah? All right. We will know all of it. So I'm going to expand the liver. Okay? This is all liver. I am now drawing it like a physiologist, apparently. So when you have the chylomicron remnant, which is what this is, I'm just going to put an X in it for remnant, because it's almost all gone, all right? Comes in. Some of it goes to dihydroxyacetone phosphate, which is part of the glycolysis pathway, and so it can be utilized for ATP. And I'm just going to put glycolysis. Okay? Whereas some other parts, the fatty parts, will go to acetyl-CoA. Okay? And acetyl-CoA has two things going on. All right? Some of it, if we need it, will be acted on by HMG reductase. And this is stuff outside of class. And HMG reductase will then make that. There's other stuff in the pathway, I believe, but this is the major thing. HMG reductase will make it into... you guys can't even see that here. All right. We'll make that into HMG reductase. We'll make that into cholesterol. And cool thing, cholesterol. Cool thing, right here, this enzyme, so HMG reductase, is what statin drugs act on, okay? And so statins turn off HMG reductase, and they have a whole ton of side effects, all to prevent cholesterol being made. There's some cool cholesterol stuff that we can do besides statins, which I recommend. So I'm just going to put a box around this. And so that's one pathway. I'm going to go ahead and switch colors so that we can see what the other pathway is. So some of it goes to cholesterol. Others will go to something called malonyl. Malonyl. And all of those are supposed to be lowercase. Malonyl-CoA. And malonyl-CoA yields fatty acids, okay? And then from DHAP, dihydroxyacetone phosphate, you can get glycerol if you are wanting to make more of, because this is going to be making the LDL and the VLDL and the HDL. I guess I should say VLDL and HDL. LDL comes out of VLDL. So right now we're just making VLDL and HDL, okay? And so if you're wanting to make more of those, then rather than breaking this down for energy, you'll keep the glycerol and maybe even make more through the dihydroxyacetone phosphate pathway. And combining fatty acids and glycerol, you first make monoacyl glyceride. All right, and then you'll add that's after you add one fatty acid, then you'll add another fatty acid and another fatty acid, and you'll eventually end up with a triglyceride. And then you use those triglycerides along with, you use those triglycerides to make VLDL, okay? So if you want to pause and go over, okay, the two places they go, and then what two things acetyl-CoA can do, making cholesterol, and then going over here, and then this green pathway from malonyl-CoA and glycerol to VLDL, if you want to. All right, next, we are going to go to lipoproteins now, because there's no time like the present for understanding what deep fried chicken does to your arteries. So, all right, so coming out of the liver here, we then have two things made at the same time. We're going to talk about one of them right now, but I'm going to mention the other one. So we have VLDL, and HDL, which right now is empty because it hasn't picked up any cholesterol. We're going to talk about the story of VLDL first, and HDL we'll come back to in a little while. Okay? So VLDL, boom. When it is made, it has the apoprotein B100, and then the same other two apoproteins. Yes, there are other varieties. These are the most important ones. Awesome. So we have ApoE and ApoC2. And all of those three are on there. B100, ApoE, ApoC2. And VLDL goes around and does pretty much the same thing as Chylomicron. It visits cells, and they get triglycerides. And it's the ApoE binding, as I recall. So let's go ahead and put this inside of a bloodstream. All right. So what happens when it starts to get low on lipids? When it gets lower on lipids, it becomes known as IDL. And then IDL does the exact same thing. This is why, if you remember, our professor said that IDL sometimes some people are like, whatever. The reason people say whatever is because IDL is the exact same thing as VLDL. It just has a different lipid concentration. And eventually IDL, after doing the exact same job as VLDL, binding with those lipoprotein lipases. Eventually it becomes LDL. And so this is low density lipoprotein, this is intermediate density lipoprotein, and this is very low density lipoprotein. And I might have these two flopped.
Oh.
I do. LDL and then IDL. No, I don't.
Oh, no.
You're right. It is IDL and then LDL. Okay, forget you saw that. All right. So LDL is when it, and so this is intermediate, as in between VLDL and LDL. The other ones are talking about density. All right. So LDL then begins picking up cholesterol, or, oh man, I'm saying stuff. It begins depositing cholesterol. Okay, so it, as I recall, this wasn't in the lecture exactly where the cholesterol came from, but as I recall, it goes and picks some up from the liver. And then it has a whole bunch of cholesterol, and it starts depositing it at cells. And so this one, I'm going to go ahead and use a different color just to make it clear that now we have cholesterol being placed in the cell by the LDL. And that has a particular mechanism which we will go over in a bit, or I encourage you to pause and go look it up, because right now I can feel that my brain is not going to be able to dredge that up. So after LDL has done this, it will return to the liver. That is a liver with an extra stripe, just for good measure. The LDL then goes to the liver and is recycled. All right? So HDL is going to do the opposite of LDL. It doesn't deposit cholesterol, it picks it up. Okay? And so it gets more and more cholesterol. This is HDL here. All right? So it keeps picking up cholesterol where it isn't needed anymore. And then it takes it back to the liver as well. Both of these, LDL and HDL, are going to be metabolized, and their parts, a lot of the parts are going to be excreted in the bile. Okay? So, and that is the entire pathway. So part 1 going through the GI tract, part 2 is chylomicrons, and then part 3 is just going from VLDL to IDL to LDL, and then HDL coming in cleaning up a bit. All right, hopefully that helps, and cheers.